As the leader in human milk science, PROLACTA Bioscience adheres to the highest safety and quality standards in the human milk industry. We are the first company in the world to create a complete line of neonatal nutritional products made exclusively from donated human milk. With increasing evidence supporting the clinical benefits and cost-effectiveness of an exclusive human milk diet for the most fragile infants, hospital demand for our products is growing globally. In response to this growing demand, we now operate 2 manufacturing facilities for the production of our human milk-based products. Our 2 facilities are situated 10 miles apart in Southern California. The physical separation of the 2 facilities provides an extra level of quality and safety because donated milk is received and quality tested in one facility, and only milk that has passed that testing goes on to our 2nd facility for manufacturing. This practice reduces the risk of cross-contamination. As hospitals continue to transition to providing the most fragile infants with an exclusive human milk diet, we will step up production to meet their needs with human milk-based fortifiers, a human milk caloric fortifier, pasteurized donor milk, and ready-to-feed products, all free from any cow's milk or cow milk-based proteins. Creating our human milk-based products starts with our donors. Each potential donor undergoes a medical and social screening. We also arrange for an at-home visit by a phlebotomist to take a blood sample. The blood sample is sent to a national laboratory and is tested for HIV 1 and 2, HTLV 1 and 2, hepatitis B, hepatitis C, and syphilis. With an accomplished scientific team from the human blood plasma industry, PROLACTA has modeled its industry-leading safety criteria for breast milk donation and testing after standards for plasma. Once qualified, donors are sent storage bags, gel bricks, insulated coolers, and prepaid overnight shipping labels so that frozen milk can be safely shipped to our Duarte facility. At our Duarte facility, milk from a cooler is pooled into containers and samples are collected for testing. Testing begins in our quality control lab with a patented DNA matching process. A DNA sample is extracted from the donated milk and is then matched to the DNA profile of the donor, which is created during the donor qualification process. This step ensures that the milk received is from the qualified donor. PROLACTA is the first company in the industry to take this critical quality and safety measure. All raw donor milk is then tested for adulteration, B. cereus bacteria, and common drugs of abuse, including nicotine, marijuana, and cocaine. We also test for prescription drugs, such as opiates. Then, the raw milk undergoes nucleic acid testing for the presence of HIV 1 and 2, HTLV 1 and 2, hepatitis C, hepatitis B, syphilis, Zika, and tuberculosis. Once milk has passed all tests and verification steps, the qualified milk from approved donors is transported to the City of Industry facility. At City of Industry, the qualified milk is first thawed and pooled into large tanks. The manufacturing process for all our products, except for PROLACT HM, begins by separating the milk into skim milk and cream. Once separated, the skim milk that will be used for our PROLACT Plus and PROLACT RTF products is concentrated to meet the target protein level. Some of the separated cream is then added back to the concentrated milk to achieve the proper caloric level. Next, predetermined amounts of minerals are added to the concentrated milk based on the specific product recipe so that when the products are used in hospitals, the final mineral content meets the guidelines published in the American Academy of Pediatrics Pediatric Nutrition Handbook. The concentrated milk product is then pasteurized to ensure the highest level of quality and safety. The final pasteurized products are bottled in containers made from BPA-free plastic. Once products are bottled, samples are selected to go through final microbiological testing for aerobic count, Bacillus cereus, E. coli and coliforms, Salmonella, Pseudomonas aeruginosa, Staphylococcus aureus, yeast, and mold. Finally, a full nutritional analysis is performed. When testing data have been reviewed and verified by the quality assurance team, the lot is approved and released for labeling with color-coded labels that include the nutritional content, the lot number, and the use-by date for that lot. The bottles are then secured with a colored, tamper-resistant neckband for an extra measure of security. Product is then returned to the freezer until it is released for commercial sale. When products are ordered, they are packed with dry ice in insulated coolers and shipped to hospital units all across the world. At PROLACTA Bioscience, our mission is to develop clinically proven high-value products derived from human milk. Our one-of-a-kind pharmaceutical-grade facilities and commitment to continued process improvement and manufacturing innovation are a testament to our dedication to quality and safety as we continue to advance the science of human milk for the benefit of patients.